How are you all? I hope you had a great weekend. Easy, Sam Roberts. We already listened to you. So the end of the road. This is, welcome to our exploration of the mollusks that's going to start this week and characterize the entire week. We're going to start out talking about International Women's Day, and I'm even going to back that up even earlier and say I hope that you had a good weekend and did some things that were based around you and not around your school or your work. Took care of yourself, took care of others. We celebrated what's likely to be in the greater Guelph area in the Royal City, maybe the last of the wintry weekends. But in the sunshine, we were able to get all of the family out on the rink in the backyard, have a fire, and we made poutine and had turkey burgers. It was not so bad. We celebrated well. But we're here now. And so I've got a couple of things. The first thing I want to do is I'm going to save this beautiful construction, and I'm going to show you what it actually looks like. This is a drawing by a particular artist and scientist that we're going to talk a little bit about today before we jump into the mollusks. So you can holster your stylo for a second, and you can see that's what it looked like. So actually, in truth, not so far off. Well done you. Put your right hand in the air. Pat yourself on the back. Emily, I think that was a lot of your drawing, so well done you. And now, what I'd like to do is, as we start out this day, we're going to acknowledge, as we already did, that it's International Women's Day. And there's a couple of links. If you haven't ever heard Kimberly Crenshaw talk about intersectionality, take 18 minutes later today and watch this TED Talk. It's super. And there's also a couple of other links from a Natural History Society in Edinburgh in Scotland, where they've made virtual issues available to celebrate and to re-educate lots of people who don't know these things. The critical role of lots and lots of women scientists in the formations of ecology, evolution, and physiology. This volume is particularly about ecology. Excuse me. And what I'd like to do before we celebrate contemporary is to celebrate somebody that you should know about, but I want to ask you a question first of all to find out whether you do know about Marion. Is that a name that you have heard before? I see, and you can see because it's on the shared screen, that we have a small disparity going here. So, I like it. I'm going to fix this. We're going to talk about Marion today. So there you can see about 81% of you had not heard of her. And this is kind of astonishing and disappointing because what an incredible person, what an incredible scientist who kind of redirected how we understand ecology and particularly entomology. And she did it 475 years ago. Wow. So she was an artist and a naturalist, born in Frankfurt, Germany, and at an era when most people lived only to an average lifespan, an average European lifespan at that point was 35 years. So I would be, as you would think of me, a very, very old person right now. At 52, she and her youngest daughter, Joanna, who was at that point 20, got on a ship and sailed to South America and stayed there for the next 21 years. And she did that because of a lifelong fascination with what Europeans called at the time summer birds. And these were butterflies and moths. And what European science, what European natural history at that point how they would have explained summer birds is that these winged creatures, ephemeral creatures of the summer, emerge from the mud. And this was done largely because of a tight tying between what you were allowed to observe, how you were allowed to interpret what you observed, and a religion and a society intimately tied up with religion, because the idea of metamorphosis and shape-shifting was inherently an evil thing. And she didn't buy this. She was a naturalist who observed the world around her. She did this from a very, very early age. Her father was an artist who kind of promoted her her artistic and her powers of observation from a very early age. And she, when met with this criticism from the male-dominated European society of the time, was like, well, she probably didn't say this, but she was bullshit. How can you not see that which is in front of your face? So she knew better. And she made a lot of critically gorgeous drawings that we can, to this day, associate the species of butterfly and moth with the species of plant that they're eating. And she tightly tied packages. She said, this is the adult, this is the summer bird, but it comes from this caterpillar, and these are the instars, or the stages. She didn't use the word instars, but these are the stages of caterpillars as they get bigger and bigger and bigger. This is what a cocoon looked like. This is what an egg looks like. Her observations let us understand parasitoids in an era when if shape-shifting of a single species was kind of a forbidden fruit, certainly understanding one species that emerged from another, a la aliens in a contemporary analogy, would have been, let's just say, discouraged. So she was the first ecologist. And so I have an invisible cap on my head, and I doff the shiny cap, or the shiny head, the cap on the shiny head, to Maria Marion, and I encourage you to read more about it. In the links that I'll post in the chat at the end of the lecture, I've got a bunch of articles, including things in Scientific American and some other places where you can read about her. She is truly fantastic, and if you have kids, this is a book, a kid's book called Summer Moths, and our kids love it, and we're going to read it tonight. So, next up is just the observation, of course, that this is not a process of celebration where we concentrate on history in 475 years ago. And here's a bunch of Twitter accounts of some active women in Twitter, active scientists, just kind of, I want to do like a kick. These are some kick-ass people that if you're on Twitter and you're interested in following some other kick-ass people, check out these women. So, mollusks. And now for something completely different. We talked, I'm going to show you a couple of slides that we ended last, what would it have been, Friday, say, with. We talked about the fact that we're going to be joining mollusks, and there are multiple competing hypotheses within this group. Now, you're familiar with this so far. We've talked a lot about competing phylogenetic hypotheses when we spent a lot of time with the ctenophores and understanding who was the earliest branching animal lineage and the consequences that that hypothesis has to our understanding of the nervous system and the through gut and diplo and triploplasts and yada, yada, yada. To Chaetognathe worms, to Xenoturbella and the mystery of the purple sock. But in each of those cases, one of the things that characterized that phylogenetic hypothesis punch-out was the fact that in some cases we didn't even know the groups to begin with, and then either remotely operated vehicles on the bottom of the ocean taught us about them, or like the Osidax worms last week, or genetics taught us about to see the things that clearly could see each other or not see each other that we were not seeing because we're not of the same scale, physical scale, and because we see the world visually with a certain restricted set of wavelengths of radiation, whereas much of the world doesn't see it at all or isn't restricted to our wavelengths, but it smells and it tastes it. But in the case of the mollusks, they're kind of exciting because these groups here, the Solanagas, the Caudofates, the Polyplecopherons, the Monoplecopherons, the bivalves, the Scaphopods, the Gastropods, the Cephalopods, forget lions and tigers, oh my, mollusks, oh my. We've known about these groups for a long time. These are good groups. We understand that and part of that has to do with this apomorphy of the group, that they produce really good shells, and those shells fossilize. And so these groups are real things. Our understanding of how they're related to each other is still changing as we add more evidence, as we discover new species, as we make new natural history observations of watching them and learning from them in situ, not in formalin or in alcohol as preserved dead things. All right, I keep working the door. Remind Smith, keep working the door, letting people in. So, this is the most conservative view, Sigward and Lindberg, the Swedish philosophical, there is nothing to see here, kind of view that there is a massive polytomy. We can do a little bit better than that. And your text does in chapter 13, which is a giant chapter about mollusks because there's a lot to understand about mollusks. So remember, as you're kind of dealing with that vast morass, that screed of a chapter, follow along with the figures that I present and orient yourself based on the lectures, and then secondarily amuse your bouche with the text. So there are three kind of big groups that we have: Aculopherians and Aplecopherons, and the Conchopherons. And the groups within and the relationships between, while there are still unresolved polytomies like between the Monoplecopherons, the gastropods, and the bivalves, there are some branches that we have an increasingly good phylogenetic understanding of that we can augment that kind of dour Swedish view. So here's some, all these groups, actually maybe some of the worm-like ones, the Solenogastra, the Aplecopherons, you might not, so welcome, and here you go. We're going to start your introduction. But bivalves, gastropods, you've known them, you've probably eaten them. Cephalopods, you love them, you've probably eaten them. Scaphopods, you might have seen them. Polyplecopherons, and there's one group actually not here, and that's kind of a cool story that we're going to get to on Wednesday. And the Polyplecopherons, or the many shields. And that's one of the first taxa that we're going to jump into today. But after we talk about apomorphies of this group. So you've done your reading for your lab this week, and you're going to be talking about and building models of different mollusks. So you know some of these things. So we'll just kind of go through the characterization of these together before you can use it as a precursor for your lab. That there is a dorsal mantle with a secreted chitinous cuticle and calcareous spicules, a shell. There is a radula, and the radula is the rasping tooth organ that we're going to talk about. That is how most of them eat, certainly how the ancestral mollusk likely ate, but lost in some groups. They have a foot. You've seen this if you can picture a slug or a snail kind of inching along with paired pedal retractor muscles. They have bipectinate gills, a tetraneural nervous system with a pair of pedal nerve cords and a pair of visceral nerve cords. Let's augment that with some images. So I've retained this image, this figure with all of these kind of morphological features of the mollusk, and it is not in your Brusca text. This is a figure of basically a generalized mollusk that doesn't exist. From Rupert, Fox, and Barnes, the dearly departed dead people. You can see on page 472 of Brusca their similar one. I like the simplicity of one, so compare and contrast these figures. But in this basic simplified version of a mollusk, we're going to talk about some of these features and realize that when we talk about this generalized mollusk, we're talking about something that's benthic, that's marine, and is characterized by having radula. So that's this is kind of the anterior end or the head. So that's this rasping organ here, that there is a mantle that's secreting a shell. There is the shell, ta-da, and there's a large pedal foot, ta-da. Now, as I said, you know these things. You've eaten them, you've watched them, you've inadvertently crushed them as you walked around campus. This is a particularly abundant snail on our campus. This is actually a European snail called Sepia nemoralis. And so the scale that we're looking at there is you can see little bits of lichen and growth on the wood. This is a very small thing, right? But of course, and sometimes I think particularly if you have, as I do, a terrestrial bias, you think of mollusks as being relatively small things. And it's important to realize that that's not always the case today, nor has it been the case in the past. This is a picture of a cephalopod, of an Ammonite cephalopod. This is in BC in one of the national parks, montane national parks, low elevation as you can see in BC. But this is an Ammonite. So this is a curled shell cephalopod that would have been cruising around the seas of likely the Jurassic, Triassic period. And so this would have been, if you think calamari, imagine calamari, that's a squid. So this is a relative of the squid that with a shell that would have been cruising around at that point. That's roughly, 4.5 feet tall. Isn't that amazing? I think that's amazing. I'll check the chat in a second to confirm that you think that it's amazing. So this is your figure 13.15 from your group that talks about the mantle or the pallium. That's a synonym for the mantle is pallium. And this is a portion of the body wall that sits over top of the mass of the body. And this mantle epithelium is what secretes the shell. And so you've got different layers. This is a cross-section of the shell. So the mantle down here secretes the epidermis secretes these nacreous, prismatic, and periostracum, the layers. And so these are various with spicules and protein, so with calcium carbonate and protein, that creates the hard mass of the shell that protects most mollusks, and particularly kind of the ancestral or ideal or this imaginary kind of classic mollusk that we're talking about. Now, some of the other, showing you pictures of some of the apomorphies for the group, we talked a little bit, and we'll elaborate this in the next few days, this tetraneural nervous system. And this is the fact, and you can see a little bit of it in these slides here, that there is a pair of pedal nerve cords. And so from this cephalization up at the left-hand side of the image, where the nerve rings and essentially the brain and the concentrated part of the nervous system are, there are two pedal nerve cords that extend down into the foot and then posteriorly along the foot. And then there's a pair of visceral nerve cords that higher up into the body do the same thing. And so this is a tetraneural system. And we'll talk about some consequences of this alignment as we go. But you think about one of the things that, particularly with gastropods, with snails and slugs, there's one consequence to this, and this is how they deal with a feature called torsion, which is a twisting of that visceral mass. We'll talk about torsion more on Wednesday and next Monday. But this is, if you trace these ganglionic nervous systems, that we have a cerebral, a pleural, pedal, parietal, and you can see, if you watch these lines, you take a close lean into your screen, hold your coffee away from your keyboard, and you'll see the lines of these tetraneural nervous systems. They cross. And that's because the body wall in the body length, we're not talking about the long, elongated kind of oligochaete or earthworm style elongate body. This is a body where the mass has turned on top of itself. And there's a lot of consequences to how they breathe, how they excrete, and how the nervous system accommodates that. But that's still to come. And you've been reading about it already as you prepare for your lab. Now, one of the things about the mollusks is their complicated and gorgeous bipectinate gills that are housed within the mantle cavity, which you'll see in a second as we go back and keep consulting with that classic kind of view of the body's the body plan of the mollusk. So these are bipectinate gills that look similar to bird feathers. So these Tenidia, which is you saw with ctenophores, is derived from the Greek for comb, that is comprised of these leaf-like filaments that extend off a supporting axis here in the center. And the filaments are ciliated. That helps propel water across the surface and sort out food particles, and at the same time bringing oxygen in. There's monopectinate, this is bipectinate characterizes this group. So that's off of this supporting axis, there are two rows of filaments. And they have what's you've run into before, though everybody has, at least in 1070 last year, we talked about countercurrent flow in the physiology section being an adaptation towards lots of things living in cold temperatures, where they use, where the venous and arterial systems are aligned and kind of laid in parallel, so that the cold water returning from maybe the feet of the goose standing in the very, 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 very cold water is warmed by the arterial blood, the warm arterial blood that's heading back out to replace it in the foot, so that by the time the cold venous return gets back to the body, it doesn't shock the system with how cold it is. A countercurrent system. You're familiar with it. And in this case, it's derived, or it's used by mollusks to have an extremely efficient oxygen extraction system. So they can live, we talked about this ancestral mollusk living benthically. One of the things that characterizes benthic waters is the nearshore waters is kind of oscillations in how much oxygen is there. And so many organisms that live benthically are exposed to tides, as you can imagine, and because of that will have to live in water that isn't at an ideal oxygen, highly saturated oxygen level. And so they'll need to be very efficient in extracting oxygen from waters that don't have a lot of it. So there's these lateral cilia. And here's a view, a graph, as you move along the gill, that the length of that branch of that pectin or the filament of the gill, you can see that the oxygen concentration in the water goes down and the oxygen concentration in the blood goes up. And that's because of this layering here of the water, that the blood, as you can see in the bottom figure of this kind of one view of one gill, is that the water and the blood are aligned at opposite directions to create this countercurrent flow. And that's housed in the mantle cavity, that's back here. So these are a view of your gills, this kind of, I don't know, it almost looks like a leech sitting there, but those are the bipectinate gills sitting on their axis. And one of the other characteristics for the mollusk is this osphradium, and it's hanging in the kind of the armpit of this, what appears to be a leech there, but is the gill. And this is a sensory organ that monitors water quality that enters the mantle cavity. And so remember, in the ancestral mollusk, there is a shell that can be, as you think about bivalves, could be open or closed. Let's do that together. Open or closed. And the osphradium then would be if you've gone kind of walking by and your boots have disturbed a whole bunch of sediment. That sediment, while it might have some food in it, the majority of it might be quite damaging, particles that are going to be quite damaging and not nutritious at all. And so in that case, the osphradium could help the mollusk understand when to close the shell to protect the gills from those particulates until they've settled. The foot, there it is, with this paired pedal retractor muscles that help it close. And if you've handled snails before, you've probably even seen that, where as you disturb, if it's out kind of cruising across and rasping algae off a leaf, then you disturb it, you can watch that foot be retracted in and close off the shell opening. Now feet, the foot, amongst different classes of mollusk, look really, really different. And so one of the neat things as we move through these different groups of mollusks is we're going to talk about modifications of the shell, modifications of the foot, modifications of the gut, modification of the mantle cavity, modifications of lungs or of gills in 5 classes of mollusk. And here's a particular one. So we have Polyplecopherons, Monoplecopherons, gastropods, bivalves twice because they're delicious and amazing. And then scaphopods and cephalopods. Now the foot is most classically imagined as, to me anyhow, and so my bias to you is in the gastropod. But the foot exists in all of the other lineages. So poly and monoplecopherons, there's a creeping foot that's kind of analogous to the gastropod. But the foot in a bivalve is a very different thing. It's more of a tongue or an arm almost that helps move it around. It's a very different thing within, and similarly within the scaphopods. And then it's completely changed within the cephalopods, which we will talk about. I hear you saying more about the Radula, but let me check for a second. I don't want to get too far. I like that you always pick up snails and move them to the other side of the road so they don't get crushed. That's good. Lesmera, which one was analogous to the gastropods? So in this specific case of the foot having a creep sole or being kind of a movable creep sole or little, what's that part of the tank called? They might even call it a creep sole after mollusks. But would be the Polyplecopherons on the top here and the Monoplecopherons. Those are kind of feet that are the most similar. Scroll down. I don't think I missed. Good. All right. Onwards. the radula. So there's a great picture of this beautiful, beautiful rasping. You can, and if you've ever spent a summer as a job or kind of worked around different houses, maybe your family's house growing up, scraping paint off fences or windows. I don't know why, but that was a lot of my kind of 8 to 10-year-old job life, was scraping paint. So I look at the radula, this organ here in the bottom right, and I think that's a paint scraper. That is a paint scraper. And it is, or rather, paint scrapers are like radulas. The design in millions of years of evolution is much better in the radula than it is in the paint scraper that you might get from the Home Depot or Rona. So the radula here, we'll talk about this whole complex. You can see it's not in some, it differs from species to species, and we use it. It has a lot of kind of taxonomically rich characters there, but it can roll across and rasp up the food and break off small bits of vegetation. And so there's the radular, is going to be a better shape. So this rasping, protrusible tongue-like organ that has chitinous teeth, and also we'll talk about even some even harder than chitin. It's not found in bivalves, some gastropods, but generally, and solenogasters, which is one of the worm-like classes, but there is a radula in most other groups. And what you can see here in this diagram from the old text that I think still shows this process is how it can be protruded, that there's a tongue-like retractor and radular etractor and protractor muscles that can send it out to rasp and then the fragments into the body. So there's the radula down and lapping away. And there's this odontophore upon which the organ upon which the radula sac and the muscles and the cartilage sits. So new teeth are constantly being secreted because, as you can imagine, this just especially if you've used a paint scraper before, if this is how you interact with the world, you wear out those teeth very, very quickly. And so that's a constant matter of secreting new teeth, and the protractor and the retractor muscles kind of move the radula and the odontophore anteriorly and posteriorly. There's the protractor, there's the retractor. Now, in different adaptations in different groups that we'll see, you can see a whole bunch of different changes in this. Some are adapted, the ancestral kind of model that we understand to be for scraping, as I've described. Some are adapted, have become adapted for drilling. This is a hole in a bivalve mollusk from another mollusk with the radula that went in to extract the fleshy animal from the inside. And some look more like raptorous birds. So break up food in the same way as a raptor would, and that's in the cephalopods. And as I mentioned, the bivalves have lost the radula because of their filter feeding existence, which we'll talk about. But even if your experience with bivalves is restricted to 1070, you've thought a lot about mussels and how they might interact with the world. Now, you may have missed it. There's been other things in the news for the last little while. But radula has, we've actually had news headlines with radula in them. That one particular mollusk called the limpet was shown a couple of years ago to have the strongest teeth. Not just the strongest teeth, but their teeth are derived from the strongest known natural material. So well done, Limpets. We're going to talk about Limpets a bunch of times. So here's this is from the Interface, the physics and engineering and biology journal called Interface. So this is the radula with these teeth. Isn't that awesome? And this is an SEM or a micrograph looking now at one particular limpet tooth on that radula. And other transmission electron microscopy, which is shown here in D, is the fact that there are nanofibers of geothite that are laid down by this limpet in a protein matrix that help reinforce the truth and provide just incredible mechanical integrity when rasping over rock surfaces. And maybe that last part of the bullet is me going, haha, you're maybe sitting there going, well, isn't spider silk the strongest natural known material? So this is actually stronger than spider silk, and it's some of the strongest synthetic fibers known to humanity. So if you're interested in that kind of intersection between engineering and physics and biology and solutions that biology and millions of years of evolution can provide and we can maybe extract and use in similar ways, mollusks are going to have a lot of ideas for you. A lot. So limpet teeth. Go out and proselytize about that, the strongest known natural substance. So using these for feeding and digestion, we'll talk about a generalized mollusk here in a second, and we'll concentrate top and bottom. So this is from your text on the top, and this is looking at the gut, and there on the bottom in the red box is where the top diagram would kind of align between your two texts. So we've talked about the Odontophore and the Radula going out and rasping. What happens next? So food enters that buccal cavity, and it's carried on mucus strings to the stomach. And this is what you're looking at here. So the stomach, this pear-shaped mass or pear-shaped sac, is located in the front end and behind the head in the anterior end of the visceral mass of the body. And there's some amazing kind of, mucus is an important part of feeding in many, many mollusks. Mollusks like mucus, particularly the more sedentary ones. And there's a fecal rod, a spinning and winding up that happens of this mucus rope that contains the food particles as it is drawn in to the stomach. There is a ciliated sorting field on the anterior end of the stomach that is grooved for sorting digestible and indigestible particles. There is then a gastric shield, which is a hardening, chitin-reinforced part of the stomach to protect the stomach from abrasion. We've seen these kinds of adaptations before, convergently evolved in other things that have to eat very, very rough material, particularly, I think most recently with some of the very sedimentary annelids that live in substrate in tubes and extract their food from substrates. And then out the intestine, and the indigestible bits go out the intestine and eventually in the feces. And I apologize, I'm just staring at the screen and actually seeing what I'm kind of gesturing at. And so the orientation of these two figures is flipped, so that the mouth on the bottom is on the left and the mouth in the bottom is on the right and the top figure. But the parameters that we talked about, those principles are the same. So there's the mucus, the rotating mucus food string. I think that should almost be the name of a band, a string-based band. So if there are musicians out there listening and you're looking for a group and looking for a name, I think rotating mucus food string or the proto-styles, and then you can explain it in interviews forever as to why. There's the ciliated sorting field and the gastric shield and the intestine out the tail end. So the coelom is reduced, the body cavity is reduced, so there is open circulatory space, there's a hemocoel, and the shell is now acting as a skeleton. This is one of the first times, this is kind of the first time that we've seen this, where instead of being just a hydrostatic skeleton, just having that, the physical properties of a closed hydrostat to help support the body and to shape it, as we saw with the platyhelminth worms and all of the variety, all of the diversity of the annelids. Here we see a shell, and the shell, and so an external shell that can support the body. Now, so we have the gonad, the coelom, and the hemocoel. So these are two coelomic spaces reduced, so it's not just the big body cavity any longer, surrounding the heart and the gonad, respectively. Now, one of the things that you've already read about in preparations for your lab this week are the hemosystems that they're characterized by having an open hemosystem. And so that is described as when blood is pumped into the hemocoel, into the body cavity, and where tissues are surrounded directly by blood. The heart is arranged dorsally in the body, and the respiratory pigment for many mollusks, although there is some diversity, is hemocyanin that is dissolved directly into the blood. And unlike our ferrous-containing hemoglobin, so giving a red or roughly red hue, this is a copper-based pigment, respiratory pigment, and so the blood of the mollusk is a greenish blood. The open system of the mollusk is compared in contrast with the closed system of annelids or our own, where we have small capillaries that connect into arteries and veins, and then interact directly with a much smaller physical scale of tissue, rather than just bathing the tissue in the blood. Excretion. There's a heart-kidney complex. There's a pair of metanephridia with nephrostomes that connect directly into the coelom. Ultrafiltrate is pushed into the pericardial cavity. These nephridia are then connected to the pericardium and to the excretory chamber. So here you can see that kind of diagrammed. So into the hemocoel, back and around. And we're going to break down the excretory system of different groups in more detail as we arrive there. So as we talked already about the Asphadia, the olfactory organ or the sense organ that's connected to the gills, there's a lot of different complexities within the nervous system. There's lots of chemosensory, there's statocysts, there's eyes, there's just amazing, some of the most complicated photoreception or true image forming eyes that we know of in the natural world are found in cephalopods, so we'll talk about that in more detail. They can be gonochoric, males and females, or hermaphroditic. The ancestral condition was to see external fertilization in the ocean, and there is internal fertilization in some groups. And as we saw with, I love this video, so there is a villager larva that characterizes, is an additional larval stage that characterizes lots and lots of mollusks. Developmentally, the blastopore does become the mouth, as you would expect with the spiralians, but which we don't see. They still have a trochophore larva, so this kind of prototroch, this mustache surrounding this lectotrophic larval stage. And so the trochophore is the larval stage that characterizes mollusks, annelids, opunculans, occurrens, and many other protostomes. But in the mollusks, it's finished, or it's followed, by this villager larva. And the villager has a small foot with two ciliated lobes together called the velum. That's what I'm tracing here. And this is a uniquely molluscan larval stage. So you can see this is the trochophore, and this is the villager. The trochophore we've talked about in some detail before when we talked about the annelids, because they also tend to have trochophore larvae. But you can see for different species, the prototroch, so this band of cilia that eventually kind of migrates through development to become near the anterior end of the body. So in the next, that's our lightning fast run through some of the apomorphies for the group. Clearly, that's not going to be it. We're going to talk about each one of these groups. And as we talk about them, we're going to talk about ways that they reinforce some of these apomorphies and ways that they depart. And we're going to jump in. The first group we're going to talk about is a group that I find particularly fascinating, and not because of their diversity, but because of some of their structural adaptations and ways of living life that are just gorgeous. And these are the Polyplecopherons. Jump back to the chat for a second before I jump into the Polyplecopherons. What is geothite? Geothite is a mineral. So that's thinking about limpet teeth. Are slugs mollusks? Or no, because they don't have a shell like snails. Slugs are mollusks. Slugs are gastropods. So they are, gastropods are a derived lineage of mollusk, the snails, and slugs. And slugs are a particular lineage of gastropods. So yes, they are mollusks and they are gastropods. And I think those are the questions that I had missed so far. So I will come back. We'll talk about Polyplecopherons for the rest of the time today. Polyplecopherons, so derive that word, and it just means, I think it's helpful to derive it, not to exercise my Greek, which does not exist, but it means, so poly means many, placo means tablet, phoros means bearing. So what is a Polyplecopheron? It is a mollusk that has it bears many tablets, if you back that up. And so it has not just one overarching shell, like my imagined gastropod, but it has 8 overlapping valves or segments of shell. And these valves have a tegumen and articulamentum layers. We'll talk about that in a second. And they have these sense organs, these aesthetes, that are embedded in the tegument of each valve. And that is amazing. And we'll talk about why in a second. So there's not that many species of them. They're marine. They're called chitins. If you've kind of been exploring in tidal pools before, maybe you have run into them and not called them Polyplecopherons, but now you can add a better Scrabble word. If you were using chitin in Scrabble, now you can try and get Polyplecopheron, which is surely worth more points than chitin. So there is one particular, there is a girdle here on the outside. This is an extension of the mantle. Remember the mantle or pallium, which secretes these valves or shell segments in this case. So you can see, of course, helpfully, Brusca and friends have labeled in this line drawing of a Polyplecopheron the anterior end and the posterior end, because as I look at that, I bet you we could flip it and we don't see a difference. So this is... and then we've got these intermediate valves. One of the ways that you could tell that a particularly a molluscan biologist could tell anterior from posterior and put aside my snide terrestrial bias joke is to look at the shape of these tablets or valves going that protect the body dorsally. So the lining of these valves is made up of different kind of layers. So the tegumentum contains conchiolin and calcium carbonate, so both protein or organic materials as well as calcium carbonate. The articulatum, so these segments here, is purely calcareous and extends beyond under the neighboring girdle and into the adjacent valve. And the hypostracum, so this is purely calcareous as well, and extends underneath the articulatum. So these different intersecting valves help create, basically, if you imagine what a chevalier is a knight armor would look like. So various different kind of, not just a uniform plate that protects, but intersecting plates that allow it to move around. And one of the things that chitins can actually do, because of the intersecting layer of these tegument, articulamentum, and hypostracum, is that they can roll up. So this muscular foot, this is one of the groups that's very much like the gastropods, has enrollment, longitudinal muscles, so it can roll itself into a ball. This is another diagram, the same kind of, it's in an adjacent cell in that same figure in your text. So a good place to examine as you're going through chapter 13. But this is now flipped over. So you're essentially in the rock, looking up at the head, the mouth, and this creeping foot. They have internally bipectinate gills, and they have an inhalant and an exhalant part of their respiratory system that is arranged anteriorly, inhalant, and posteriorly, exhalant. And you might think, what a straightforward thing to call about and to identify. And this is a bit of a tip of the hat to when we get to some other groups where, in fact, the inhalant and exhalant canals are not arrayed as elegantly as this. So they are browsers. The Radula that uses different minerals like manganite or geothite is another example. Thanks for that question in the chat. They have a mucus strand. So here they flipped again. And so for some reason this slug is heading east and the mouth is on the right-hand side. But odontophore and the rasping Radula and then into the body. And so very similar looking to our hypothetical benthic mollusk, with one of the exceptions being the valves. There's the Radula, there's the stomach, there's the intestine. And chitins can in fact be not just rasping, but they can be predatory. So watch that chitin under the probe, and you see that it slaps down. So there can actually, there's a genus called the Placophorella that is predatory. So it does eat things that are smaller than itself. Now they're gonochoric, so males and females. They have a single gonad, and we're looking there at this large single gonad with these gonopores opening up into the exhalant canal and heading out so that fertilization is achieved externally. They have a trochophore larva and that molluscan, villager larva, that cute foot-bearing stage that happens after the trochophore. In some groups, we don't see it within the Polyplecopherons. They do have the Tetranorus nerval system, so the pedal cord, paired pedal, and visceral cords. They have a subradular organ, as you've seen in some other groups, so it's a chemosensory structure located below the radula. It's probably in a neural ring arranged anteriorly. So essentially, what we have is a slow-moving marine herbivore and a grazer that lacks a clearly demarcated head. And this is a view, this is actually sped up quite a bit, but this is a chitin moving across rock, and you can see that creep sole and kind of moving around and arranged out here, so not in the fleshy mantle, but here in the shell, on the edges of these valves, you'll see little bumps, and that's where I want to spend the rest of class today. The next kind of 6 minutes is talking about these bumps, because some of them are protection, like this, and some of those bumps are eyes. Hundreds and hundreds of eyes arranged around the lateral sides of the body. That's kind of amazing. There's no knight. We talked about using the analogy of saying these kind of overlapping valves as being like, this is what a knight would really want, a chevalier like I would really want, and for sure they would do it. And then it gets into like science fiction character territory if that which protects could also allow you to see. Chitins, Polyplecopherons have achieved that. So within these valves, interlocking valves forming the shell from anterior to posterior, we see a bunch of different features. And I talked about it, I give you the name already, the aesthetes already. Now these are different parts of the aesthete here. So you can have big openings, small openings, mega or micro, and they're in the tegumentum, they're in the shell. And that some of them have photosensory caps and cells within them. So these photosensory cells, we'll take a better look. This is an older drawing. But some of them are not just photosensory, they have lenses. I'm going to jump even ahead in the story and say some of them can form images. These are image-forming eyes in the shell. So not even of the shell. Let's even clear that. So they're not. It's not like they have secondarily derived eyes that kind of insert themselves in the shell. This is an eye made of shell. So there's a cornea and then an argonite, a mineral lens that has retinal cells and an optical channel. And the grains in the shell at that point are arrayed differently. And so this is one of the ways, again, the structural nature of this is incredibly important, because in the protective part of the shell, on the left-hand side, these the mineral layers inside of that shell, that compose the shell, are laid down in a disorganized fashion. And that helps contribute to strength in this case. But in the lens part of the shell, all of the manganite is arrayed. It's aligned, and that allows light to pass through and allows the same material, instead of functioning as protection, they've turned it into a lens. So here's a bunch of different kind of views of this that I'll break out for a second here. So we've got the whole animal, and we've got these non-sensory solid protrusions and the eyes embedded. There's so this is a microscope, and this is a scanning electron a micrograph, and this is a view of looking at that megastellate and looking at the micro and megastellites, and then there's a pit there in the center. That's where the eye sits. And what I'd like you to do for a second is to show me in the chat. Think about this. Why, if you're a slow-moving marine herbivorous grazer that lacks a clearly demarcated head, why so many eyes? All right? If you've already got ideas, throw them in already. Polyplecopherence could be a great monster in Monsters, Inc. Yes, I think I agree. I think that's amazing. So you're a slow-moving marine herbivorous grazer, which is not an insult. I am clearly just describing how you live life. And so already some of your ideas popping up is that to be able to spot predators and threats easily. Yes, and in fact, so combine that with another observation that enables them to see in 360 degrees, enables them to pick up images from all around you at once, Maybe find the best areas for food. And yes, if they're slow moving, they potentially don't need to use up as much energy on movement. That's an interesting kind of physiological evolutionary hypothesis. I like that. So instead of needing to dedicate energy on movement, and now you can, because you can kind of allocate it defensively and say, well, I'm not going to move fast. My strategy is not escape. My strategy is just to be able to see and maybe protect as threats arrive. And I think you're getting at a lot of the reasoning there. And oh, that's good. I have to check that out. The last season of The Mandalorian had a cephalopod shell creature with a bunch of eyes that was freaky. That's ringing a bell in this Swiss cheese brain. I have to look back at that. Okay, I'm going to jump back. I'm going to keep the chat kind of where I can see it, but I'm going to jump back to the last kind of explanations that we've got here. There's a figure in the bottom here where in red with the solid area of the shell and in blue the lens area of the shell, you can see that the lenses are actually, there is a cost to those lenses. They're not as strong as the rest of the shell. So if you were to put a load on the eye, it's going to break. And so it is providing the shell, because it has both these non-sensory solid protrusions and the photoreceptive areas. This is essentially an evolutionary strategy that gives you the best of both worlds. Where there is protection, there are these big, red bars, strong non-sensory areas. But there's microscopic lenses that let you, if you're a slow-moving browser, perceive fish and shadows, not just shadows, but even see the shape of fish. So we have integrated sensory elements as well as incredibly subtle protection that's permitted here. So from the whole animal to the eye to the protrusion. So the mechanically weak sensory regions are protected in valleys by protruding robust regions. That weakness is offset by having thick, hard underlaying layers. And then multiple eyes, and some of you talked about this in your thoughts in the chat, helps create a redundancy in case there's grazing, but also gives you the protection of having the full 360-degree panorama. So predation, even when it harms individual shell plates, or as older parts of the shell are damaged, there's continually reproducing parts of this shell. And so there's redundancy that lets you replace damaged areas and scan the entire area. This is amazing. Chitins, this is one of my favorite mollusks. Absolute favorite mollusks. And you can see some, I'm not alone in that, because there are different parts of the scientific community in some of the highest ranking journals that were really excited by this a couple of years ago. I'm going to post a bunch of links from today, from back to Marion to eye-ridden chitins, and I'm going to stop because I've gone a couple of minutes over with my enthusiasm for the eyed shell and the first ecologist. And I'm going to say, have a great lab, those of you that have your labs on Tuesday. And I'll see everybody else back here on Wednesday. And have a great lab. I'm going to answer some questions in the chat and post links.